As we continue on with our renal anatomy, we really can't get by without doing an in-depth look at the nephron anatomy as well. And we kind of alluded to this when we'd done our general urinary anatomy with the kidney earlier. But we have to investigate the nephron, look at how it breaks down regionally, because we are going to be using those regions as distinct functional points in that nephron, with each part of the nephron having a role to play. We once again have to make sure we understand how the nephron fits into our general kidney anatomy. And once again, we have to make sure we understand how the blood flow to the kidney relates to the blood flow to and around the nephron. So again, we have two general types. And as we went through our kidney anatomy, we really made note of these being anatomical differences. We have cortical nephrons and we have juxtaglomerular nephrons. Cortical nephrons stay more so up into the cortex of the kidney structure itself, with very little tubular portion extending down into the medulla. Conversely, however, our juxtaglomerular nephrons we see extend further down into that medulla structure. Now, again, this looks very anatomical, very structural on the surface, but keep in mind that this particular portion of the kidney, the nephron, has a very specific role to play with reabsorption. So once we talk about this particular portion of the nephron, you could make an assumption that one type of nephron, cortical versus the juxtamedullary nephron, could have a greater role to play with the function that goes hand in hand with this particular portion of the tubule. Functionally, we want to use our nephrons to produce a filtrate. This is going to be known as the functional subunit of that kidney. We want to take fluid and solutes from the blood and create a filtrate that now will go through our nephron tubule. Anything that we still have a use for, we want to reabsorb that back out of the nephron tubule and into the blood, allowing only true waste products and any otherwise useful solutes that we just have in excess to become part of the urine. So when we look at the nephron and we start breaking it down, we can look at it grossly as a vascular component. This is where the filtration from the blood will actually occur, and the tubular component. This is where the reabsorption is going to occur back into the circulatory system. Now, when we look at our vascular component, remember we had our glomerulus. That was our highly specialized capillary bed through which we filtered the blood. The Bowman's capsule is going to be distinctly part of the nephron. And together, both of those are going to form the renal capsule. So when we look at what we mean here, here is our afferent arteriole bringing blood into the glomerulus. Here's our efferent arteriole carrying blood away from the glomerulus. Here's our glomerular capillaries. The surrounding structure here is our Bowman's capsule. Anything that leaves the glomerular capillaries and passes through these slits are going to wind up as a filtrate inside the Bowman's capsule. And eventually, that filtrate will extend out into the tubular portion of the nephron. Together, the glomerulus plus the Bowman's capsule is going to make up our renal corpuscle. And to put that into perspective with the rest of the anatomy, here is our vascular portion. That is going to be the portion of the nephron that deals directly with the glomerulus and is directly involved with the filtration of the blood. The remainder of this anatomy is our tubular portion of the nephron. The first portion we get to is our proximal convoluted tubule. That's going to be the portion directly right after our Bowman's capsule. Proximal, if you remember our directional terms, our anatomical terms, means closer to the point of attachment. This implies that it's closer to the Bowman's capsule. The proximal convoluted tubule is going to be deeply involved with reabsorption. Remember that most of the solutes we're filtering out of the blood, we still have use for and we still want. So as soon as that filtrate leaves the glomerulus, enters the Bowman's capsule, and starts its journey down the proximal convoluted tubule, even right here and there, we immediately reabsorb up to about 80% or so of all the solutes that we just got done filtering back into the blood. Anything that didn't get reabsorbed at the proximal convoluted tubule continues on into the loop of Henle. Now we've got two distinct portions of the loop of Henle. We've got a descending limb and we got an ascending limb. The loop of Henle was the portion that would extend down into the medulla of our renal pyramid. The descending limb being the initial portion, we make this hairpin turn, and then we go through our ascending limb. The loop of Henle is going to be primarily responsible for passive reabsorption of salt and water. Purely passive. So earlier on, we had our nephrons divided up into two broad categories, cortical and juxtamedullary. We said that we could make some inferences based on which type had which function because of the greater portion of that loop of Henle. We can assume that cortical nephrons are going to be more or less responsible for reabsorption of salt and water in a passive manner, whereas the juxtamedullary nephrons are going to be more geared towards that specific function. So we left our Bowman's capsule, we entered our proximal convoluted tubule, and we immediately reabsorbed a significant percentage of all of the solutes that we had just gotten done filtering. Whatever's left that didn't get reabsorbed continued on down our descending loop of Henle, up our ascending loop of Henle, and while it was in the loop of Henle, we reabsorbed water and we reabsorbed salt. At this point, whatever still didn't get reabsorbed winds up in our third portion of the tubule, the distal convoluted tubule. And for all intents and purposes, even though they're structurally different, we're actually going to link the function of our distal convoluted tubule in with our fourth portion of the tubule, the collecting duct. They run in that sequence, so they're distinctly different anatomically, but functionally, we're going to lump those together. These are the portions of the nephron where we can exert hormonal control. You remember the hormones that we talked about when we did cardio that were responsible for blood volume regulation. We had antidiuretic hormone, we had aldosterone, we had atrial natriuretic peptide. These hormones act on the nephron, but they don't just act on any part of the nephron. They act on the distal convoluted tubule and the collecting duct in order to reabsorb varying degrees of water and sodium. At the distal convoluted tubule and collecting duct, we also see some actively secreted solutes. We'll secrete certain toxins into the distal convoluted tubule. We'll see transport here as a means of us getting rid of hydrogen ions at the distal convoluted tubule and collecting duct. But first and foremost, we're going to think about the distal convoluted tubule as a means in which the hormones are going to act. Now, keep in mind, when we say reabsorb, we have to keep in mind the vascular network through the kidney and how it relates with the nephron. When we studied the kidney, we trailed our way through the renal artery, segmental artery, interlobar artery, and so on. And when we got to here, we said these were blood vessels directly associated with the nephron. Here is our glomerulus. We had our afferent arteriole carrying blood into the glomerulus. We had our efferent arteriole carrying blood away from the glomerulus. They are both arterioles. Arterioles meaning carrying oxygenated blood, but more importantly, they have a distinct ability to constrict and dilate, both of them. And it's worth pointing out again that even though we consider our glomerulus a capillary bed, it is not a capillary bed taking on the functions of a typical capillary bed that we've been accustomed to talking about. We do not see a bidirectional fluid shift at the glomerulus. We do not see gas exchange at the glomerulus. This is a filter with a blood pressure generating a filtration pressure, pushing a certain volume of fluid and any solutes that will fit through the slits in these capillaries out into the Bowman's capsule and down the tubular portion of that nephron. Anything that doesn't fit through those slits simply carries on with what's left of the blood fluid out the efferent arteriole and eventually into the peritubular capillaries or the vasa recta, differing anatomically. The peritubular capillaries and the vasa recta, those are capillaries that more accurately represent what we knew about capillaries. This is where we get gas exchange, making up the cells of the nephron. Remember, the nephron is not a cell. The nephron is a structure composed of many cells. They need to be oxygenated. They need vascularity. So our peritubular capillaries help supply those cells with what it needs. But because of their nature of wrapping themselves around the entire nephron tubule, those capillaries will also be the site at which anything reabsorbed out of the nephron enters back into the circulatory system. So overall, we see urine formation occur along the nephron tubule. We picture the kidney as a layperson, as a filter. But the only part of that structure that really looks like what we think about as a filter, like a cauldron, is going to be at the glomerulus. We pass blood through the glomerulus and we create a glomerular filtrate. Significant amount of water and any solutes that would fit through the glomerulus. That filtrate carries on into the nephron tubule, where now, based on the different locations, proximal convoluted tubule, descending limb of the loop of Henle, ascending limb of the loop of Henle, distal convoluted tubule, and collecting duct, we see various roles in reabsorption. As we pass through the tubule in a sequential manner, we reabsorb what we still want to recoup out of that filtrate, put it back into the circulatory system. And the closer we get to the end of the collecting duct, the closer we are to refining the composition of the urine we're producing. As we're in the collecting duct and the distal convoluted tubule, remember, we have the ability to also do some secretion at that point. Toxins, certain waste products. And from our discussion, we're going to specifically bring up hydrogen ion secretion. Anything that makes it to the end of the collecting duct is going to be considered urine. And again, to tie this back in with our renal anatomy, anything leaving the collecting duct passes through that renal papilla and into the minor calyx, then into a major calyx, then into the renal pelvis, down the ureter, and into the bladder. Here's a bit more of a simplified drawing with fewer blood vessels on it. But here would be our afferent arteriole, our efferent arteriole, and anything that would be associated with the tubule is going to be peritubular capillary and or vasa recta. And we can see anything that gets reabsorbed out of our tubule wall can go right into the circulatory system at that point. So it's important that you understand the regional anatomy of the nephron, because the next step we're going to get into is reabsorption. We have to look at how we reabsorb things, because every single region that we just talked about is going to have its own distinct mechanism on how we recoup solutes from the filtrate that we just produced and put it back into the circulatory system. So if we have any questions about that, please reach out and let me know. And we'll be moving through the rest of the renal system in a very systematic way. Thank you.